Welcome back to biotechnology. Today we're going to talk about ELISA, which is a method for measuring things, allergens. I'm going to do some drawing, then I'll come back. So you're all familiar with the standard pregnancy test, or maybe not, but you'll have seen it. And it's fairly simple. It's a stick. You pee on one end. Somebody pees on one end anyway. The wee goes along, and the example, the result would be either a negative sign or a positive sign, typically, or two lines, or some less or more frivolous, less useful piece of information. But the basic system has two bits of information here at this end, one or two lines, one of them to tell us that the machine is working, and the other one to tell us that it has done the thing that we want to measure. So what is it doing? It's measuring a protein in wee, human gonadotrophin protein, and it's going to determine whether it passes a threshold amount or not. That's all it can do, because we've only got a plus-minus test. And we need to do that using some way, so we need to detect the molecule in the pee. Let us zoom in on the parts. So down here we're going to have some antibodies. Antibodies look like our university symbols, like this, and they stick onto antigens, which are proteins typically, but can be other things. Usually they're proteins, and in this case it is. They stick onto a protein and are carried along in the flow of wee. That doesn't help us very much at the moment because we haven't made it coloured, so we can't see it, and we haven't let me stop so I can get another pen. So here I am with my other pen. This is a dye molecule, so it's blue. I've just drawn it like that because I felt like it's going to go along with the wee, and we need to have it stick on to this position in two ways so that we can make the two symbols that we want to determine whether it's worked and whether we have the molecule in the wee. So what we need for that is some way for it to be stopped at that point, and we also need antibodies because they are a good way of doing that. So our antibody is a protein. It's got an amine group at one end and an acid group at the other end, so we can imagine we can attach things to those. We can attach our dye to one end. Our recognition region that's going to detect our protein is on the side up the top here, so I should have drawn it slightly canted onto the top, and that is first of all going to recognize the protein human gonadotrophin G, HGG. Then we want a way of these being captured, so they need to stop at the surface, and we can use another antibody for that, and we can either attach to the antibody, which is good. So if we have an antibody to an antibody, so this line here is anti, and it will attach to this end here. It'll attach to the end that's going to detect our substrate. It's just going to attach generally to it somewhere, and it will bond that to the surface. So we've got another antibody down here attached to the surface, and what it will eventually do is end up with one of these stacked up on it like this, one that hasn't been attached to our human gonadotrophin G, HGG, or even one that has. It doesn't make any difference because it's going to make our minus symbol, which tells us that the test is not broken. If we leave these too long, or they're stored too warm, this might break down and then it wouldn't work, and we need to know that because it's kind of important for these tests. So that would be our negative line, and our positive line would be another antibody to the antigen, so to this protein, and that will be here. So this is a normal antibody, antibody 2. We'll call it antibody 2. It needs to be a different one because we want to attach to a different site on our protein, and that will make this line, because then on this position we will have, let me check how I've got some space. So we've got the antibody, we've got our antigen, We've got the other antibody on the other side, and we've got our dye molecule on the top. This is not ELISA, this is called a linear flow device. It's the simple way. Why is it the simple way? Well, if we look at it, we can detect a certain amount of dye. So if there's a tiny amount of this protein in there, it will mostly not appear, so we won't be able to see it. If there's more than a certain amount, we can see it, but if there's a really large amount, we can't see any difference, it's just black. It saturates very quickly, so it's not a very useful test if we want to measure something that is more complicated, where it matters more if there's a little bit, a medium amount, or a lot. The antibody can be made. If we try to make an antibody to an antibody, we need to go into a different organism, because obviously it would be not very helpful to you if your antibodies attacked other antibodies. That would be super unhelpful, and so it doesn't happen very much. They usually manage to not recognize own body antibodies. There are autoimmune diseases, but they normally go wrong in a different way. They normally recognize some types of cells. So one type of diabetes, for example, is an immune disease where your antibodies are attacking your own islet cells and killing them, and that causes diabetes. There are other forms of diabetes. So in this case, we've got 3 types of antibody. 2 of them are bound to the surface by this bottom group, and 1 of them is floating in Wee. The really cool thing about these is that the amount of P that goes up them is fixed by the volume of the device. So at the top there's some kind of reservoir where it goes in. It's all done by capillary action, so it's just a piece of paper, not normal paper. It's made out of glass fiber, probably because it's easier to manipulate the surface of glass fiber, it's easier to make it sterile, and it doesn't have genetic material itself, so it's not going to interfere. We need to be able to attach these antibodies to the surface, so this one needs to be attached and this one needs to be attached to the surface. They need to be printed on in some way, and all the other places where things can attach need to be covered up so that we don't lose this on the way up, so it doesn't stick somewhere. Because usually, if you've seen one of these devices, usually there's some kind of plastic cover that covers up all of this bit so you can't see it. The only bit that sticks out is like a little bit at the end where you put the Wee. If you do a drugs test, it's much the same. It works in exactly the same way. Some drugs tests. Not all drugs tests, obviously. Some of them are more complicated than this. Stop. So getting on to our main theme of ELISA, which is enzyme-linked immunosorbent assays. Nearly got confused there. We have some kind of surface. It's typically a little well in a plastic plate, a 96 well plate typically. 96 because it's easy to form an array out of them. If we put attach this to a surface, then let me start again. So we have an antibody attached to the bottom of our well, and we're going to apply our antigen, which is this green blob here, which just in the case, exactly as in the case we just had, the linear flow device, it will stick to the recognition site of the antibody. That's the antibody's normal job in solution, but we are now using it for something different. We're using it to recognize the antigen. These antibodies aren't 100% effective. They sometimes recognize the wrong thing, but this type of assay is going to do 2 attachments, and that's much more effective. So first of all, we start off with this. We may have to apply that in the first step, so we may take our dish, and we may have to attach this. So we wash it through. It has an attachment point on the bottom, and it attaches to the bottom of the dish. That's a possibility. Or we could buy it already coated. Then we put in our antigen, and then we put in a second antibody from a different animal, which also recognizes the antigen, and this is called sandwich ELISA. As you can imagine, we've sandwiched the antigen, which is a protein in this case, between 2 antibodies. Now we've effectively linked this blue thing to the surface through our molecule that we want to test, and we can now detect it. Now we can use the enzyme. So in the example before, we had a dye on the end, and that would work fine. But an enzyme is better because an enzyme can make lots of product. So if this was the typical enzyme, which is horseradish peroxidase, it can make a blue dye by breaking down a precursor molecule. So we put in the precursor molecule, it makes the blue dye, and we can detect it. But it has an additional advantage. If we leave it for a longer time, at a slightly warmer temperature, it will make more blue dye, which means that we can detect a lower concentration. The problem with our linear flow device was that above a relatively low concentration, it tops out, goes black, and then nothing else can happen. Once you've covered all of those sites, even if you use a super duper machine to measure, it still doesn't make any difference. With this, we can effectively count molecules. If we get down to too low an amount, just one of these that happens to manage to stick somewhere non-specifically will cause our result. So we obviously have a limit on the lower end of our detectability, but it is a single number of molecules, and like a countable number of molecules. On the upper bound, if we've covered all of our surface, so if we filled all of these up, then we can't measure any more, but we could dilute our solution. So now we have a system where we can measure more by just giving the enzyme longer, and we can measure lower concentrations, we can measure higher concentrations by diluting our sample, and because it's outside the body and it's in a 96 well plate, we can do all of that all at once. So if you watch pretty much any news program, eventually you will get a picture of somebody doing a pipetting into a 96 well plate, and that is exactly this. Typically the result will make a fluorescent dye, because fluorescent dyes are relatively easy to detect, or a coloured dye. But fluorescent dyes are even better because they're easy to detect. You can detect them in extremely low amounts, and that's because of the physics of fluorescence. We put in a light that is blue, for example, and we get green light out, so we can make everything black and no green light comes in, and then it is comparatively easy to measure the green light because we have no disturbance. We're not subtracting anything from anything else. As you will know from physics, if we subtract numbers from each other, we get error, whereas if we don't have to do that, it's a lot easier. So onto a specific example. If we want to measure, for example, food, and we've got some nuts, for example, that we think might be hazardous to our health. Nuts get a fungus in them, and it makes a type of poison that is extremely poisonous and gives you cancer. That's obviously not a good thing, so we would like to know that and take that out of the food chain, out of the supermarket, wherever it's in. We need to get it back out again, so we might test food that contains nuts. We want to test it for the toxin. It's called aflatoxin, and it's a protein, so it is detectable in this way. So we take our aflatoxin, we put it onto an aflatoxin detecting antibody. It attaches, we attach a second antibody to the top, and then we can measure the amount of aflatoxin in there. In that case, the amount that's allowed is extremely low, so we don't need to worry too much about the detection stage. We need to worry about our concentration stage and measuring accidental false positives. But because the toxin is extremely bad, we need to worry less about measuring the exact amount. In my case, I have an allergy, so we can consider that case as well. It works slightly differently, and my allergy is to wasp stings, and the wasp sting protein that gives me an allergy is an antigen, and I generate antibodies. So what they really want to measure with this is how many antibodies do I make to see whether I'm likely to die the next time I get stung by a wasp. So how do we find that out? We take our antigen, which is in my blood, and we attach it, the antigen, we know the antigen, that's the wasp sting protein. We take the wasp sting protein, we attach it to a surface, so instead of this we have the wasp sting protein. Then we take my antibody, effectively my blood with the red blood cells taken out, because that's kind of red and nasty. So we just take the white fluid from it and we add it to the top. The antibodies go and attack the antigen, they attach to it, because that's what their job is. That's why they're causing me to be allergic to the poison in the first place. So obviously if they don't do that, that means that I'm cured anyway, so it is self-correcting. So if my antibodies now attach to these proteins, these wasp sting proteins on the surface, then we can use a second antibody, an antibody, which we know we can do because we had one in the flow cell, in the flow test, in the linear flow device. Sorry about that, that was moving so fast the video couldn't keep up. So if we have an antibody, so it would be a human antibody from some other animal, then we can attach an enzyme to the top and we can measure how much I have. In that case it's important because we will get a number. The typical numbers in that case go from 1 to 5 from an older test. It's actually called RAST, from a radioactive detection method, but it's not done like that anymore. Radioactive sensors, radioactive tracers are very efficient, so if this had a radioactive tracer on it, we could measure it with a piece of old style photographic film really easily. You just lay it on top, leave it for some time and have a look. If it doesn't detect, then you get another piece and leave it on there for longer, so we have the same advantage that we had with the enzyme. But the disadvantage is we generate radioactive waste. It's not very high level waste, but it's a headache and it's expensive to get rid of and causes problems. So we don't do that very much anymore. So it's a risk, it's a safety risk, and it's expensive. So it's better not to do it, and this method is way better because it's safer and we can throw away the waste. This is all biomolecules. The only thing that might not be biodegradable is the plastic here. Everything else is just biomolecules. We can just throw it away in the compost. What else do we need? Yes, there's an important step, however, that I missed out, which is rinsing, of course. If we have some antigen and we put it on our surface, we do need to rinse it extremely well, because if any of our, particularly this molecule, gets stuck somewhere, it will be measured and we will detect it anyway, and that's not very helpful. The problem there is usually non-specific binding, that our antigen or our antibody can just stick to something, just generally. In the example that I've done in the lab, the protein that we put on the surface sticks by hydrophobic interaction. So that is relatively simple, it's easy to do, but the problem with hydrophobic interaction is it's completely non-specific. If we use an antigen, we don't know which way up it is, so some of the molecules won't be able to attach. Whereas this way around it's quite efficient, because the bottom end of the antibody, this third attachment point, is different to the other two, so we can use the chemical difference to attach it specifically in the right direction. Unsurprisingly, to bring us up to date, this is exactly the technique that was initially used to detect coronaviruses, because what we would then do is have the antigen stolen off the virus and detect the antibodies from the victim, from their blood. Obviously that doesn't tell us whether they have an active infection, but it does tell us whether they were exposed to the virus at a reasonable time in the past, which is also helpful information. Okay, that's enough for today, because it's a nice topic that can be closed like this. So I'll leave it there. Cheerio.